Last time we talked about Mendelian inheritance, and now we're going to talk about little alterations from that. We're going to talk about sex influence, inheritance, and some other things like gene mapping. So we'll start with sex influence traits. And get it to work. Okay. A famous scientist named Thomas Hunt Morgan in the turn of the century was working with fruit flies. Genes were a very new concept at this point. But the reason why they're working with fruit flies is because they're, have quick generation times. In a matter of days, you can make these fruit flies, and they can lay some eggs, and then they hatch. You can look at their phenotypes under a microscope, and we'll be doing some of that in lab. What he noticed was that he had male flies, some of them with a white eye, and usually the eyes are red, but he didn't find any females with this trait. So what he did was he crossed a mutant male allele with, so the ones that had white eyes, with a female that has red eyes, and he found that all the F1 progeny, males and females, had red eyes. So he concluded that this was a dominant trait. He then crossed his F1 females with the ones with the red eyes but had been mated with the white-eyed males with F1 males. And what he found was that he did have expression of the white, but only in males. All the females were red-eyed, and then most of the males were white-eyed, but some of them also had white eyes. Only males had white eyes. What he did, a test cross with F1 female, so breeding it with a homozygous recessive, and then doing some other F2 generations. He found that they did have white-eyed females, that they were viable. It wasn't something lethal to the females. And from this, he concluded that, okay, it must be something with the sex chromosome. And that this was a trait that was on the X chromosome. And the reason why that is significant is because the X and the Y chromosome are different in size. A Y chromosome does not have as many genes as an X chromosome. You can see in this picture here, the X chromosome is a lot larger. So there are a lot of genes here on this X chromosome that aren't on the Y chromosome. And in fact, if in humans you only have a Y chromosome, that will not develop into a full human and will usually end up in a spontaneous abortion or miscarriage. But females with only an X chromosome, with only one, are actually able to survive. So the inheritance pattern of this sex-linked trait did not follow the same pattern as the Mendelian traits that we talked about last time. So to illustrate this, I'm going to do a Punnett square of a trait on the X chromosome. Okay, so these are two eyes. This is our phenotype, and we're going to use color blindness because that is actually something that is on the X chromosome. And what we have here, this is our female. Okay, here. And this is our male. This is the symbol for female and male, if you didn't know that already. Okay. A female has, as we know, two X chromosomes. And a male has an X chromosome and a Y chromosome. And a Y chromosome is almost like having nothing at all. Okay? And what we're portraying here is this male is blind, meaning he has, and for color blindness, we're going to call it the R. Okay, R.
But actually, we're gonna make it true. So little r. Okay? And this female, not colorblind, but she is heterozygous. So she has a big R and a little r. Now, the Y chromosome does not hold the gene, only the X chromosome does. So we use a little different notation. Rather than using just these letters, we actually use a subset. So we use X little r to denote the allele on the X gene, and then he has a Y. And then so this female would be X big R and X little r. Now if these two then mate, we can determine the probability that their children will be colorblind by doing a Punnett square analysis. So if I'm going to put the male on top here, this will be his genotype, the X little r, and then he has a Y chromosome. We can also determine from this what proportion of males are females. Okay, so the female is on the side here, and she has X big R or an X little r. And then we just fill in the Punnett squares just like we did before. Same thing here. Okay.
Avast virus database has been updated.
Sorry about my database. And now, I can ask questions. We can answer questions about the probability of their children having colorblindness. Oops, this is wrong. Oops, wrong. So this should be X big RY here. That one was fine actually. But this one should be X little r y. So the first thing we can ask is, what's the probability that they will have a boy or a girl? Okay. To add up the probability of girls, we just say, okay, this one has 2 X's, so it's going to be a girl. This one has 2 X's, so it could be a girl. So, and then there are 4 possible, so 50%, 2 out of 4. Same thing for males, 2 out of 4, 50%. But if I ask, what's the probability that you will have a male and he is colorblind? Okay, this is the colorblind because it has the little r here and the Y, which makes him male, and because the Y does not have the gene on it, then it's basically like having nothing. So you could almost just erase this Y completely when we're talking about the colorblind allele. Say, okay, that one X is only what's going to determine the allele. But we know that, it does have a Y, but we'll just draw it smaller. It has a Y chromosome, but it's smaller, so it doesn't have the allele there. So what percentage of them will be male and colorblind? Well, 50% will be male, so 1 out of 2. And then of those males, 50%, this one, will be colorblind. So the probability of getting a male that is colorblind would be 1 in 4. But the probability of, if you are a male and being colorblind, would just be 1 in 2. We also have this female is also colorblind because she has 2 recessive alleles. The probability of being female and colorblind is the same as well. Anyway, from there we can determine then what the different genotypes of our sex-influenced traits can be, specifically on those on the X chromosome. Alright, so a little more about the X and Y chromosome. In females, you have 2 X chromosomes. Males, you have 1 X and 1 Y. I already said that. Humans have a total of 46 pairs. 46 total, 23 pairs. 22 of those are autosomal, and 1 pair of the sex chromosomes, and that would be the X and the Y. Y is a lot smaller. In other animals, They have a similar, some of them have similar, like birds have a similar sex chromosomes as us, except they are opposite. Rather than the heterozygous being a male, the heterozygous is a female, and the homozygous is the male. I think grasshoppers or honeybees are a little bit different as well. So, like I said before, an X-linked recessive allele, one's associated with an X chromosome, will affect males more than females, and that's what T.H. Morgan found. Some of those are red-green color blindness. So if you cannot see the number in here or the shape in this one, then you are probably have some degree of color blindness. This is the color blindness test. So in here is 74. If you couldn't see that, then you're probably colorblind. And in here is a shape of a sailboat. Hemophilia is another a blood disease which people have that doesn't have a clotting factor, so when they bleed they can't form a scab and they can keep on bleeding, so their bodies can't prevent themselves from bleeding. Heterozygous females do not have symptoms of this. They're asymptomatic. So it is a dominant recessive, but it's on the X chromosome. And this is a problem in royal families in Europe, and it was introduced by Queen Victoria of England, who was an asymptomatic carrier. Another phenomenon with the X chromosome is called dosage compensation. And what happens is females have two X chromosomes, but usually one of them is inactivated. And if the two X chromosomes have different alleles, and because this is done at random, this will give different expression in different parts of the body. So you can see this in the coloring of some cats, where the X chromosome that expresses brown pigment is expressed in some, and the chromosome that expresses black pigment is expressed in others, and so it gives it this brown and black splotchy appearance. And then another gene is controlling the non-expression, which would be white. What happens is one of those X chromosomes is inactivated and condensed, and it becomes what's called a bar body. So all females are mosaics, meaning they are partly expressing something in some parts of their body and partly expressing something in the others. There are some modes of inheritance that do not follow chromosomes at all. So, for example, mitochondria and chloroplasts have their own genes, and they are expressed or passed on usually through the mother, which is called maternal inheritance. But depending on the sex, it could be passed through the father as well, or depending on the species. Now, until now, we've been talking all genes are chromosomes.
Or different chromosomes. But some genes are on the same chromosome, so they are not following the laws of independent assortment and segregation. And to be able to determine how close genes are on the same chromosome, scientists have done recombination, or have looked at recombinant expression of genes. And we will do some of this on the worksheet later. But the basics are, if they are together, then on the same chromosome, then they will be expressed together. But if they are expressed differently, then an event must have happened to separate them, which would have been crossing over, which occurs during synapsis and meiosis. Alfred Sturtevant was a scientist who figured this out, that the actual distance between two genes is correlated with the expression of recombination in progeny, which is what I just said there. So if you have a 1% recombination, he actually quantified this as being one math unit, which he then called a centimorgan, naming it. I don't know if he named it or if someone else did, but 1 centimorgan then is a distance between two genes.
And you can use two-point test crosses where you're looking at two traits to look at then those differences. But if you look at three different traits at the same time, you can then determine what the order of the gene is, which way they go. And we're not going to actually determine distances by three-point test crosses, but we will determine the order in the worksheet we'll work on. So one thing humans have done with the genetic code is to look at where genes are on each chromosome. And this has historically been done by looking at pedigree, so looking at human parents and the occurrence of alleles, including diseases. But it's been hard because those actual disease-causing alleles are actually quite rare. But once anonymous markers or things that you can actually go in and tag different sequences of DNA were developed, it made it a lot easier, and you didn't have to actually have a phenotype to figure out what the gene was. Single nucleotide polymorphisms, or SNPs, are differences in DNA, looking at different individuals, based on just different nucleotides or different base pairs. And this increased, again, the resolution of mapping, because now you can find the difference between actually two individuals. And this is actually what is done to determine, the paternity of a child. So if anybody's seen those talk shows where they're determining the father of a child, this is what they do. They send it in for an analysis. They look at the different base pairs. And based on those base pairs matching up with the child, we can determine if they in fact inherited genes from that person or if it was someone else. Sickle cell anemia was the first disease, and we talked about this in lab a little bit, but to be a known result of a mutation to a protein. And this causes a defect in hemoglobin, which disables it to carry as much oxygen, and also gets caught in blood vessels. But heterozygotes for sickle cell anemia have some normal shape and have some non-normal shape. And they are able to actually, for the most part, live out a normal life, not experience any of the symptoms. Sometimes some of the symptoms may happen, but for the most part, they are fine. What was found is that the prevalence of sickle cell anemia is mostly in tropical areas because sickle cell-shaped blood vessels also prevent contraction of malaria, which goes through blood vessels, a little protozoa that infects blood vessels. So it has an advantage of preventing malaria, which is a very devastating and prevalent disease in these tropical areas, which has lots of mosquitoes that also carry the disease and spread it from one person to the other. Alright, another thing we mentioned in lab is non-disjunction. This is where homologous pairs fail to separate. Aneuploidy is when you have a gain or a loss. Monosomy is when it's lost, so now you only have one chromosome. Trisomy is when you have an extra one, so now you have three. You can see this is trisomy 21, which is Down syndrome. You can see this chromosome has 3 instead of 2 is what it's supposed to have. So in these small chromosomes, like in chromosome 21, if you have a third pair, that's usually fine. But in these larger ones, it will usually end up in, again, a miscarriage or a non-viable, non-developmentally viable human. If it happens on the X or Y chromosome, it's generally has severe developmental abnormalities, but some of them, however, can reach adulthood and can even be fertile. Triple X females are usually have an extra X chromosome. XXY is called Klinefelter syndrome. XO is Turner syndrome. I mentioned before, if you just have a Y chromosome, then those you will not survive. And XYY is called Jacob syndrome. Another thing that has to do with how things are inherited is called genomic imprinting. and that's where the phenotype or the expression depends on which parent contributed the allele. So, for example, Prader-Willi syndrome and Engelmann syndrome both have to do with chromosome 15, but if it comes from the father, it's Prader-Willi, and if it's Engelmann, it comes from the mother. And here's two examples of this is Prader-Willi and Engelmann. You can see there are differences in the facial structure of the different syndromes. So genes, however, are not the whole story. We do have some things called epigenetics, which is the things surrounding genes, including histones, the proteins, which actually, well, the genes surround them, and the turning off and on, which happens through a process called DNA methylation, where different groups, molecules can be attached to DNA to turn them off or on. And this can change then the chromosomes and the structure and what's expressed, and can also be passed from mother to offspring. This is a new area, relatively new area of research that has a lot of influence on the health and expression of genes in humans. So one thing we can do to detect genes, we can do it in vitro, which means in utero, in the uterus before a baby is born. And there are three different ways to do that. One is a pedigree analysis, so just by looking at the traits of the parents and the grandparents and so on and so forth, you can come with some probability of having certain traits. Another one's called amniocentesis, and that's where fetal cells are collected from the amniotic fluid. The amniotic fluid contains the cells similar to the baby itself, and then you can do DNA analysis on that and look for certain markers of diseases. Or chorionic villi sampling, a similar thing where cells collected from the placenta are examined for genetic traits. This is done, like I said, mostly to look for genetic abnormalities that may affect the child's development or even its ability to survive outside the uterus. So this has then led to another thing called pre-implantation genetic diagnosis, where even before, and this is connected to in vitro fertilization. What happens in vitro fertilization is a woman is given hormones for it to superovulate, where it ovulates more than one egg at a time. Those eggs are taken out of the ovary of the woman, and then in a culture, you can fertilize them with sperm, and you can grow them for a certain amount. Once they're grown, they're split into multiple cells. You can then take one of those cells and then do a genetic analysis on that. Once that genetic analysis has been done, you can then take and decide which of those fertilized eggs you want to implant back into the uterus, and then that can develop. So it's usually done to screen, again, for genetic diseases, but could also be used for, if you wanted to figure out the genes, for hair color, for height, for IQ, whatever you can find a gene for, you could screen for in this process. So it has some ethical concerns. Is this really ethical to be able to pick and choose what genes you feel are important or able to be passed on? And there's a little video on, if you want to click on that, you can look at this video on PGD, or pre-implantation genetic diagnosis. Alright, that's it. Again, we will work on some more practical applications of this on our worksheets, and I hope you had fun learning about genetics.